Good morning and welcome, everybody. How are you guys doing? All right. Today what I'd like to do is explain a little bit about how molecular motors work in the context of microtubules. And once again, to sort of motivate this sort of question and idea, I show this picture of a neuron drawn to scale. So this is a mouse motor neuron, and is just a little bit of a overview of this slide. We've got the neuronal cell body here with some short dendrites. We have a synapse that would be here, and this would be the type of neuron that would be found in a mouse that would be connecting the spinal cord down to its foot. And one of the really dramatic things that you see is that most of the neuron is actually made up of axon. Now, a big problem, at least theoretically, is explaining how proteins made in the cell body are transported down the length of the axon. And when people have done careful mathematical analysis of this problem, it turns out that it would take years, if not decades, for materials just to simply diffuse from their site of origin here in the cell body down to the synapse. And the other thing is that there would be a really dramatic concentration gradient. So in diffusion, what happens is that there's a really high concentration of materials where stuff is produced, and then that drops off with distance. But here we have a problem where stuff needs to be uniformly distributed along the length. And these problems suggest that there's some active transport mechanism, and it turns out that that mechanism involves kinesins and dyneins, and that's what we'll talk about today. And in addition, we'll start to introduce intermediate filaments with the sort of goal of eventually building up an understanding of neurofilaments, which are important for the structural integrity of axons. Okay, so this is just reminding us of these topics that we're going to talk about today. And just in terms of a little bit of a review, here we have a cartoon of a neuron. We have the neuronal cell body here. This is the nucleus of the cell, a cartoon of a dendrite and an axon. And we recall that microtubules have a polarity. They have a very dynamic plus end that undergoes dynamic instability, where it elongates, has a catastrophe, rapidly shrinks, there's a rescue, and the cycle sort of goes back and forth. But in any case, in neurons, the plus ends of microtubules, for the most part, point in the same direction along the length of the axon, and that is towards the synapse. And one thing in terms of transport is that this information about, or this polarity along the length of the axon in terms of the microtubules, helps establish the polarity of the axon itself. And this goes back to an idea that I introduced a couple of lectures ago, that the proteins that make up microtubules have a polarity, and then that creates polar structures, and then when cells are built of polar structures, they have a polarity. So these scales of things just build up. So another part of transport to remember is that transport is bidirectional. So it's essential that new materials are being transported and moved out along the length of the axon into the synapse. And some of these different cargoes in terms of transport involve organelles and vesicles. So for example, mitochondria, mitochondria in particular, but also precursors for synaptic vesicles and whatnot. So these move out at a very fast rate. In addition, there's these cytosolic proteins we've talked about, actin and tubulin. These are transported at a much more slow rate. And then in addition, there's stuff that needs to be transported back to the cell body. One part of this involves lysosomes. So lysosomes are involved in the degradation of different organelles and proteins. So when material needs to be degraded in the axon, it gets packaged up into lysosomes, and then that gets transported back to the cell body. But in addition, there's also signals, for example, like neural growth factor needs to be transported back to the cell body. And for these anterograde pathways that move stuff out and these retrograde pathways that move things back, there's two different classes of motors. So kinesins are going to move things out, and dynein is going to be moving things back. And we'll talk about these right now. So this is a picture showing the structure of different molecular motors that are known to be important in cell biology. I will say, first off, these aren't quite drawn to scale. It turns out that dynein is a massive molecule. It's about 3 or 4 times larger in terms of molecular weight than kinesin. So it's not quite to scale, but you can kind of get a feel for the different structures. At the top here, we have Kinesin 1. So this was the first kinesin that was discovered. It has these head domains, a short little neck domains, and then kind of a long neck or stalk. And then there's some white chains that bind to the tail that are important for associating with cargos. There's a couple of different other types of kinesins. So there's a kinesin 2 that is really important for transporting cargos in cilia and flagella. There's also some involvement of transport in neurons. There's the kinesin 3 class of families, which includes UNC104, and that type of kinesin in particular is important for the transport of synaptic components from the cell body out to synapses, obviously. And then just to kind of contrast, so the point here is that I'm saying is that there's a big class of different kinesin families, and this is just a small subset. And each one of these kinesins has a different transport function and also different functions in the cell. In addition, here's myosin 5, which we talked about when we were going through these laser tweezer experiments. These are the two heads. What you can see is that myosin 5 has a very long neck domain, and then once again, cargo binding domains, and then finally this is dynein down here. And we're going to go through these in a bit more detail in a bit. In terms of the kinesin superfamily of proteins, years ago, people sort of randomly named kinesins a lot of different things, and there sort of reached a point where there became a little bit of confusion as to what different kinesins were called. And at one point, there was a nomenclature that was established where you have things like kinesin 1, kinesin 2, kinesin 5, kinesin 13, that very systematically organizes the different types of kinesins. As I had sort of mentioned before, so kinesin 1 was the first discovered, in part in the lab of Michael Sheets, who I had done a postdoc with. So it in particular is important for binding to different organelles and transporting it along microtubules. And here's kinesin 2 down here. There's another type of kinesin that's really important both during the process of mitosis and in axonal elongation. That's kinesin 5. And it's kind of interesting in that it has a sort of overall structure that's very similar to non-muscle myosin 2. And that is that it forms these little sort of filaments where there's motors on both ends. And in various sort of different contexts, it can cause microtubules to slide relative to each other. So in our muscles, actin and myosin work together to cause actin filaments to slide. In the mitotic spindle, kinesin 5 works with microtubules to cause microtubules to slide. So there's two sort of parallel systems for generating forces. Very often, actin generates contractile forces that pull things in, and very often microtubules generate extensile forces that are pushing things out. And part of the rationale for that is that cylinders or tubes have a lot of structural integrity, and they can handle strong compressive forces, where if you have an actin filament, which is a helix, a rope-like braid, it can handle strong tensile forces. So two different types of physics. One handles compression, the other handles tension. two different types of motors, sort of parallel functions, but things going in opposite directions. And then finally, this is Kinesin 13, which I often say is very unlucky for microtubules because it can bind both to the minus end of a microtubule and the plus end of a microtubule and drive disassembly. And part of the way that that works is that the kinesins usually walking along the microtubule, but that Kinesin 13 actually grabs the tubulin subunits and pulls them back, and it causes the ends to splay out. And when that happens, microtubule disassembly follows. So different classes of kinesin. So you can see we've got Kinesin 13 here. We've skipped a bunch. There's a lot of kinesins that have very specific roles in mitosis, and we'll talk a little bit about those in a future lecture. So Kinesin 1 is the general purpose anterograde motor that moves cargoes towards the plus end. And it's just a reminder, so anterograde means to move things outward, retrograde means to move things back. Like non-muscle myosin 2 or just myosin 2 in general, it has a head domain. Instead of binding actin filaments, it binds to microtubules. But like myosin, kinesin also hydrolyzes ATP as an energy source. And that is what is powering the transport of these cargoes. So short little neck domains, a long stalk here, and this is sometimes called a coiled coil domain. So you have a coil, two different proteins interacting. And then once again, we have our kinesin light chains at the very tip of the tail to interact with cargoes. In terms of the mechanism of how kinesin walks, there are similarities with myosin in general, but some of the details differ a little bit. One really important way, well, I'll just sort of walk through this. So essentially what we have here is kinesin, and it's bound with ADP. It's going to bind to the microtubule, and then it will release that ADP. That'll be exchanged with ATP. During this process, it causes a conformational change that causes one of the heads to swing forward, and then ATP hydrolysis follows, inorganic phosphate is released, and then we're back to ADP, and the cycle can repeat. So conceptually, kinesin has a very similar mechanism to myosin in the way it walks. And like if you were to watch a cartoon, it would look very much like somebody walking. So kinesin's just taking steps along microtubules. The one sort of key difference, and actually one of the keys of discovering kinesin, is that when ADP is present, Kinesin 1 is not locked down to the microtubules. And one little trick to the initial discovery of kinesin was that there was an analog of ATP called AMP-PNP. So this is phosphate, nitrogen, phosphate, which is a non-hydrolyzable analog. It can't be hydrolyzed, which means that it can't be broken down. So AMP-PNP has an overall structure similar to ATP, it just can't be broken down. And it turns out that when AMP-PNP is mixed with Kinesin 1 and microtubules, it causes the kinesin to be locked down to the microtubule. And in the initial process of discovering kinesin, one of the approaches was to take microtubules, mix in AMP-PNP, Kinesin 1 binds, one can spin those microtubules down and form a pellet. The kinesin's locked to it. You can remove the AMP-PNP, spin the microtubules down again, and then you have a relatively pure fraction of Kinesin 1, which then would subsequently be much enriched and whatnot. But this is just sort of, yeah, a little bit of a history of the problem. So, in terms of this mechanism, so Kinesin 1, it's binding to cargoes, it's moving towards the plus end, and the sort of complementary motor to Kinesin 1 is Dynein, and this is a cartoon of that. Much like all of these other motors, there's going to be a microtubule binding domain. We have some stalks, and then we have these head domains, and then there's going to be a cargo binding domain that's attaching with other proteins. Like with myosin, like with kinesin, dynein utilizes ATP as an energy source. And in terms of its mechanism of action, it's similar in that initially we start off with dynein bound to a microtubule, ADP is present, ATP is going to bind, it gets released, there's a conformational change, ATP is being hydrolyzed, and then inorganic phosphate being released, and this causes the power stroke of dynein, which allows it to move. And here's sort of, well, these are electron micrographs showing the structure of dynein, and we've got the stop domain up here, and then there's this big head domain, and a big part of the mechanism by which forces are generated is that ATP hydrolysis causes this sort of conformational change. And I guess it's a bit subtle, but can you see that this sort of has a curve thing, and then during the power stroke, this donut-like structure rotates, and then that's what's actually causing the microtubule binding domain to move forward. So dynein by itself is not processive. So when we were introducing the idea of myosin, we have processive and non-processive motors. So processive motors can bind to a filament, and they can take multiple steps, and they can move things over long distances. Non-processive motors bind, generate a force, and then they release. Dynein by itself is non-processive. So if you just mix dynein in with microtubules, it doesn't really transport the organelles very far. And one protein that helps it is dineactin, and it helps form this complex to move things along microtubules. So this is dineactin sort of all together here. And I'll just sort of mention this. There's two really important domains. There's a microtubule binding domain and a cargo binding domain. So dineactin doesn't generate any force. All it's doing is it's taking a cargo, it grabs the cargo, it's grabbing onto the microtubule, and it's tethering the cargo to the microtubule. What happens next is dynein is also interacting with this complex, and dynein is generating the force. So dineactin is helping the cargo stick to the microtubule. Dynein is generating the force that's moving things along. So dynein and dineactin are working together as a complex to transport cargoes. In terms of some of these different structures, I'll just sort of point this out. So we talked about ARP2-3, so actin-related proteins 2 and 3 being important for branches on actin filaments. This is actin-related protein 1, and is involved in the formation of this dineactin complex. And there's a couple of other proteins. P150 glued is a really important part of this sort of like dineactin complex, and actually that's what people would consider dineactin. So altogether, these molecular motors work to organize the distribution of organelles in the cell. So once again, we have our microtubule organizing center in the center of the cell. The minus ends of microtubules are associated with that. They're being nucleated by gamma tubulin, which is enriched here. The microtubules extend outwards, and in particular, so for lysosomes, what happens is that as lysosomes mature, make a transition from being late endosomes into lysosomes, dynein interacts with this, and it's moving stuff back towards the minus ends of the microtubules. Here's the mitochondrion. It has kinesin and dynein, both associated with this organelle, and this allows mitochondria to be transported out to synapses where they can generate energy. You also have things like the Golgi apparatus associates with dynein, and then that actually causes the Golgi normally to be close to an MTOC. And one of the big points is that in terms of neurological diseases is that disruption of axonal transport is thought to be involved in a lot of different diseases like Alzheimer's, Parkinson's, Huntington's disease. We're not really going to get into the details of that, but there's definitely higher level courses that cover this in depth. So with that, that's the review of the molecular motors, yeah, so it's in some ways just providing the foundations of what you need to know about molecular motors. And I think what you'll see is there's a sort of a basic theme. You see that with myosin, you see that with kinesin, you see that with dynein, of head domains binding to filaments, using ATP to generate force, and then cargos being transported as a result. So the next thing I'd like to do is talk about intermediate filaments, and the major point of intermediate filaments seems to be to provide structural support inside cells. And this is an image of a non-neuronal cell where intermediate filaments have been labeled up. We have the nucleus of the cell here, and you can see these sort of strand-like things, mostly in the interior of the cell, that are just sort of forming a meshwork. It turns out that these intermediate filament proteins are built up from dimers, and here we can see that we've got this N-terminus and a C-terminus, and it basically forms this helix here, and this is the primary building block. And I'll just ask you, or point this out here, does this protein have a polarity?
Yes.
Yes. So there's a distinct end over here that is going to be different than over here, so that it has a polarity. So the interesting thing is that these dimers then make tetramers, so you can see how they form. And I'll ask, does this have a polarity?
Overall, no.
Exactly, right. So this does not have a polarity. And I'll tell you, these tetramers are the basic subunits that make up intermediate filaments. And just to sort of walk for anybody that doesn't get that, if you look at this, you have an N-terminus and a C-terminus. And if you were to flip the protein around, it would flip the entire protein around. It would change where the N and the C-terminus are. If we go to this, what we have is a C-terminus and an N-terminus. And if we were to flip that upside down, we would still have a C-terminus and an N-terminus. So this complex, when you have two polar dimers coming together, they can make a nonpolar structure. So the question that I'm sort of getting at is, why is this important? And we'll sort of lead into that. In any case, these tetramers assemble to form protofilaments. So here's a protofilament here. So you can imagine there's going to be a little tetramer there. It's going to form a protofilament. And then these twist together to form these protofibrils. And then these come together to form these intermediate filaments. So the question here is, if the subunits are nonpolar, they don't have a polarity. Are the structures themselves going to have a polarity? Are intermediate filaments going to have a polarity?
No.
Okay, exactly. So if the subunits don't have a polarity, the structures they build up will not either. So intermediate filaments don't have a plus end. They don't have a minus end. And this is one really important difference between intermediate filaments, actin filaments, and microtubules. There's a couple of other implications here. So with both actin and microtubules, this polarity is really important for the mechanism of the motors. The motors need to know which way to go to generate forces. So if you put a motor on a filament that doesn't have a polarity, would the motor be able to, well, what would happen actually? I'll ask that.
It shouldn't be able to stick to the surface.
Let's say that you do have the motor, it can stick to the surface, but the filament's not polar. So, yep, go ahead.
It wouldn't move. I feel like it would be lost, like it wouldn't know where to go.
Exactly. So it wouldn't move, it would be lost. It would act like it doesn't know which way to go. And in fact, that's entirely correct. So you could imagine that without a polarity, a motor might take a step in one direction, maybe it'll take it in the same direction again, but then randomly it'll go the opposite way. So without polarity, you would just have diffusion of those motors where stuff is like being moved back and forth, but no long distance transport. So the intermediate filaments, they lack polarity, and it turns out, at least at present, there's no known molecular motors that interact with intermediate filaments to generate forces. And that's part of that sort of implication there. But in any case, if you look at the structure, it looks very much like a braided rope. And once again, coming back to this idea that a big role of the cytoskeleton in cells is to provide structural support, this rope-like structure, once again, very good for handling tensile forces, so stretching, keeping cells together. So one of the big themes, or one of the things that's important in terms of understanding microtubules and actin filaments, is that they are dynamic. They use ATP hydrolysis to control their assembly and their disassembly. It turns out that intermediate filaments do not associate with ATP or GDP. But nonetheless, they are still dynamic. It just turns out that they're much less dynamic than the microtubules and the actin filaments. Just to kind of illustrate the dynamics of intermediate filaments, so this is a cell that was injected with a type of intermediate filament protein called keratin. In fact, keratin is one of the proteins that makes up our hair and our fingernails and whatnot. So if you see somebody with really pretty hair, you can tell them, you have beautiful keratin, and they would think you're a geek, of course. But in any case, this cell has been injected with these subunits, and about 20 minutes after injection, there's these tiny little spots that are showing up in the cell. And this is showing us that there's a little bit of formation of intermediate filaments. If you come back at 4 hours after injection, well, actually, just to explain this figure, this is showing the keratin intermediate filaments as a total, as a sum. That's showing us everything. This is biotin labeled keratin. So biotin is a molecule that binds to avidin with high affinity, and very often people use avidin, biotin to cause things to come together. It's a useful sort of molecular tool. So what one can do is biotinylate keratin and then use some other tools to come back and find out where the keratin is. Using this biotin is kind of a hook. So these cells have been injected with keratin, conjugated with biotin. That starts to get incorporated into the filaments. After 20 minutes, there's a few dots. After 4 hours, you can start to see that there's a pretty good overlap between these two things. And the implication is that even though ATP hydrolysis and GTP hydrolysis aren't important for the dynamics of intermediate filaments, they're still able to polymerize and depolymerize. They just do it a lot more slowly, and they do it much more like just sort of dumb polymers. So like when people make polymers for plastics and whatnot, the way that things assemble doesn't have a polarity, doesn't have an energy source. Keratin intermediate filaments much more like that. So, okay. And as I sort of mentioned, there's a lot of different types of intermediate filament proteins. So the keratins, which I had mentioned before, they're associated with epithelial cells, really important for tissue strength, structural integrity. There's other types of intermediate filaments that associate with glia, muscle, mesenchymal cells, and once again, providing structural integrity there. In neurons, intermediate filaments are called neurofilaments, and there's three major types. There's a heavy neurofilament, a medium neurofilament, and a light neurofilament. One thing that I think is a little bit interesting is that while in humans and vertebrates, neurofilaments have been shown to influence the diameter of the axons, when neurofilaments are lost or disrupted genetically, it turns out that it doesn't have a huge impact on the development of animals. So you can knock out neurofilaments in mice, they still develop. It just turns out that some of their axons have a smaller diameter than they would otherwise. And there's some other sort of subtle phenotypes involved. In addition, there's invertebrates such as, well, flies, things like crabs, lobsters. These types of animals don't have neurofilaments at all in their neurons. So there are intermediate filaments that are still present, but these specialized neurofilaments are absent. So this sort of suggests that while neurofilaments are important for providing the structure of neurons, especially in our body, they're not absolutely essential for the formation of a neuron, an axon, a growth cone. So still involved, but not central to this process. And then finally, sort of continuing this theme that intermediate filaments protect, there's intermediate filaments called lamins that surround the nucleus of our cell, and they build a little cage that helps to protect our DNA. And this is sort of showing us that, so here's our non-neuronal cell. We have the nucleus of the cell here. You can imagine the nucleus of the cell is going to sit right there, and it's going to be surrounded by this meshwork of intermediate filaments. And lamins, in particular, are just going to be right around the nuclear envelope, holding it together. The very interesting thing is that defects in laminin can lead to this disease, Hutchinson-Gilford progeria, which is a premature aging. And actually, I'll just throw this, any guess on how old these two gentlemen are?
7.
Yeah, they're about, they're teenagers, so yeah. And what's interesting is that in this particular case, there have been mutations in the lamins, and as you can sort of see, they look very old, and they have, people with these sort of mutations have a lot of symptoms associated with the elderly. So there really is premature physiological aging that's going on. Now, it may seem really weird. So intermediate filaments, you disrupt an intermediate filament protein and it causes one to age faster. And sort of the puzzle is to why. And one thought that is out there is that we have DNA in our nucleus, and normally our bodies are moving around, and when they move around, occasionally that's going to damage or break DNA. And that is leading to the premature aging. With those lamins in place, it is protecting the DNA in the nucleus, so as we move around, the DNA is better protected, and this slows the aging process. And that's the sort of theory to explain this particular sort of disease. I don't know. I think it's very interesting. It sounds very plausible and stuff like that. But yeah. Okay. And then I guess just to sort of summarize things. So over the last couple of lectures, we've been talking about microfilaments or actin filaments. These bind to ATP. They form these sort of meshworks. Tracks for myosin, really important for force generation. We've talked a lot about microtubules, and we've just introduced intermediate filaments. So I guess with that, I'll just ask if there's any questions. So, okay. Other than that, I'd say have a great day.